Sathyabama Institute of Science and Technology.
Good morning, everybody. This is Jesia Persis Preethi from Department of Biotechnology, Sathyabama Institute of Science and Technology. So, if you have to speak about genetically modified organisms or even cloning, we need to mention cloning vectors. So, what are cloning vectors? Cloning vectors are specialized organisms in which we can incorporate the DNA that we desire, and using this DNA, we can replicate it into our desired quantities. So, cloning vector is a specialized DNA sequence that can enter a living cell and provide a means for detection of its presence to the researcher by conferring a selectable property on the host. And if you have to speak about vectors, the physical traits of the vector, that is such as size and shape, has a very important role to be played. So there are n number of cloning vectors, out of which I'm going to discuss about four important types which had been used widely. So first and foremost, plasmids, which can uh, in plasmids we can incorporate up to 10 kb of desired gene of interest, and viral vectors we can incorporate up to 30 kb. And coming to the other one, cosmids, we can incorporate up to 35 kb, and artificial chromosomes up to 100 kb we can incorporate. If you see a cloning vector such as plasmid vectors, so plasmid vectors are nothing but extra chromosomal double stranded DNA which is present in predominantly in prokaryotes, and these plasmids, size of the plasmids generally range between 1 kb to 200 kb, and we can efficiently incorporate up to 10 kb of our desired gene. So if we have to speak about the three important regions which are present in the plasmid vector, one is the ORI site, which is the origin of replication site, and the lag gene which helps in using the galactose. For example that I've used, I'm using the galactose gene, and the next is the uh, antibiotic resistant gene. So these are the important sites which are required for which are present in the plasmid, which makes it useful for our desired need. And we also have to mention about the multiple cloning sites which are present on the plasmid, which can help in incorporating, which can help in splitting apart the DNA and incorporating the gene of interest. So in the example that I've given, you can see there are different nucleases which are mentioned. So there are different cloning sites like ECO-R1 and SMA1, BAMH2. All these multiple cloning sites are present, which can be used very effectively to incorporate the gene of interest. So how do we prepare these plasmids? How, in order to get these plasmids from the cell, we need to subject it to various phases. First and foremost, the cell wall which is present has to be disrupted. So for a gram negative bacteria, it is easier compared to a gram positive bacteria. We need to subject it to, we can choose the method of our interest. Either we can physically physical methods, or mechanical or chemical methods we can adapt in this context. So once the cell wall is disrupted, we can get the content of the cell which is present out, and this cell content is centrifuged to separate the DNA, RNA and protein and the cell debris. So the DNA, RNA and protein is present in the supernatant, whereas the cell debris are present in the pellet. So by using different enzymes and different chemicals, we can effectively separate the DNA. And for this purpose, we also have to speak about the surfactants and detergents that we can use for this process. DNA and SDS phase comes of effective use. So for the extraction of plasmid DNA, we subject it to treatment with EDT and lysozyme in the presence of sucrose, which will prevent the cells from bursting forth, rather forming spiroflasts. And these spiroflasts have cytoplasmic membrane intact. Therefore, when we subject it to Triton X100, we can remove the cell extracts, and once the remnant is centrifuged, we can separate the plasmids from the cell debris. So this is how we can effectively separate the plasmids, and this plasmid can be used for cloning the gene of our interest. So thank you.